for me, that would be helpful. So your name, first and last name, just so that when I save everything, I have a little bit of a record of who's here, who's not here, that sort of fun stuff. Thank you guys. Okay. Good morning. Okay, so on Fridays, we're going to meet virtually. On Fridays, we're going to meet virtually. You guys all found the room. I apologize about the announcement. I had typed up a better one yesterday, and I thought I had hit post at like 1 to post it at 1 p.m. yesterday, and it did not do that. So I don't know if I picked the wrong date, if it will post next year or something super fun, but I apologize. So in the future, I'll make sure the announcement actually goes up. It'll be the same link. It's a recurring meeting, but I'll post the link every week just so that you have it accessible to you. Okay, so again, Fridays are going to be our journal club. This week, I'm not doing a typical journal club. I'm just going to do an overview of some of the methods that you're going to come across when you do your reading, because for a lot of people, reading a scientific paper can be intimidating, in part because the methods can be a little bit foreign, right? In class, we talk a lot about these concepts and ideas and biology and physiology and what's happening in our cells, but then a paper comes through and they don't really explain a lot of stuff very well. So I wanted to give everyone kind of a foundation for microbiome research as we think about our gut microbiome and the effect it can have on our body. Next week, I will present a paper. Let me pull out that paper. I'll talk about this more next week as well. I am a crazy person. I don't like to read on screens, so I kill trees and I print things out and I make like all sorts of notes, and I have like different colors that mean different things. And this is just the way that I find I can read papers most effectively. If you are like me, feel free to print things out. If you're not like me, just read it on a screen. You gotta figure out a system that works for you. So next week I'll do more of a traditional journal club. And then starting on the 26th, then we'll start having our student presentations, and we'll do 2 people a week. Again, at some point in the semester, you're going to present and you're going to write about 2 people's presentations. So if you miss a week, that's okay. Just try to, at least when you're present, like be active. Anthony, I'm going to mute you. There we go. Okay. So today I'm going to talk a little bit about microbiome research. If at any point something doesn't make sense, please, please, please stop me and we'll go back through it. Okay, let me share my screen here. Okay. Can you guys see this? Can I get a nod from some of the people whose camera's on? Can you see this? Awesome, thank you. You know, the fun thing about doing a Zoom class is you have no idea like what's happening. Like who can see what, who's paying attention. Anyway, okay. So your homework was to talk about sequencing technology, was to watch a short video about sequencing. Now, I'm not sure who has had genetics or not had genetics, and how comfortable you guys are with sequencing. So I wanted you all to watch that video so that we're on the same page about sequencing, the old way of doing things, right? Sanger sequencing. And what today's class is going to be about is high throughput sequencing technologies. And this is something that you're going to see over and over again in the papers that you read when you're thinking about the gut microbiome, because microbiome research, a lot of it is based on sequencing. Okay, so our learning objectives are to compare Sanger sequencing with next generation or high throughput sequencing, to explain how you can identify different species in a community using next generation or high throughput sequencing, and to detail the workflow for a high throughput study. So keep in mind, when we get to our first test, it comes from the learning objective. So I'm going to go through a fair amount of material here, but as long as you feel comfortable with these 3 things, then you should feel comfortable with this lesson. So again, if you are able at the end of this to compare and contrast Sanger DNA sequencing, which is what you watched that video about, with next generation sequencing, if you're able to explain how you can identify species and detail that workflow, then I would say that you're in a good place with this material. Okay. So let's first talk about the history of high throughput sequencing. And I am a nerd, so I find this stuff to be like super interesting. And I think it's really important now when we think about it, because you've been hearing a lot about sequencing on the news, right? All this talk about COVID variants, that comes from sequencing technology and the speed and cost of sequencing technology. Back in the day, we started out genomics really, with the Human Genome Project. This is where they wanted to sequence the genome of several model organisms, okay? And this was put together largely by conventional Sanger-based sequencing. So think back to that video about how Sanger sequencing works, okay? How Sanger sequencing works. What they detect is different strands, right? So in Sanger sequencing, you have these special nucleotides that end the strand building, and they have a fluorescent marker usually, right? And so you add a small amount of those, and as the strands are replicated using traditional PCR type technology, right? Certain ones are going to end with those fluorescent markers, and then you can run gel electrophoresis, and you can get them by size, and you could read through, usually with some sort of computer program, the different colors for the different sized pieces, and that will tell you the sequence, okay? That's the traditional Sanger sequencing. We're detecting the end, the termination of a strand. Starting in about 2005, we started to see the appearance of several new sequencing platforms. And this is where things started to get different, okay? So we called Sanger sequencing, that is chain termination sequencing, And that, again, is we are adding a special nucleotide that will stop the formation of that chain, and that's what we're detecting. High throughput systems do something different. And instead of doing chain termination, they detect the release of a pyrophosphate when a nucleotide is added. So rather than waiting and measuring all the different ends of sequencing, we're sequencing by synthesis. And this will make more sense when I show you guys some videos, okay? This is called deep sequencing or next-gen sequencing, next generation sequencing. All of those terms, high throughput, deep, and next gen, they refer to the same kind of technology. Okay, this graph I find like really exciting. Again, I'm a nerd. High throughput sequencing. So this is showing you the year up until about 2019, and this is showing you the cost per genome. So back when we did the Human Genome Project, we spent like $100 million on it, right? Keep in mind, this scale here, this is a log-based scale, right? Math, this is a log-based scale. We spent a ton of money on that first genome. And Moore's law would say that there's like a simple log relationship here, and we would expect this sort of depreciation in cost. So we would expect 2019, so, you know, a year and a half, 2 years ago, we would expect it to cost like $100,000 to $250,000 to sequence a genome. And in fact, it costs about $1,000, maybe even a little less, right? That is crazy. So in 20 years, we have gone from $100 million to about $1,000. And this is part of the story when we talk about COVID too, is that we're able to do these sort of sequencing experiments so much faster and so much cheaper than we used to be able to do them. And it's allowed us to do things like create vaccines and detect variants and all of these things that we're talking about in the news. We're able to do it a lot faster, right? So the speed is just remarkable. This high throughput sequencing is going to be commonly used for sequencing genomes and transcriptomes. These omes mean the whole. So genome is all the genetic material. Transcriptome is all of the transcripted material, so all of the RNA, right? So genome, we're looking at DNA. A transcriptome, we're looking at RNA. We're not talking about the cost for like a small plasmid. If any of you guys have taken genetics, right? A little plasmid, a little teeny bit of DNA, a little bit of DNA in a little circle. We're not talking about that. We're doing all this stuff. It's very exciting. This requires you to make a library of DNA fragments. So again, you're going to take all of the DNA, you're going to chop it up, and you're going to make something called a library. And each platform that we're going to talk about varies in terms of how that sequencing is performed. Okay, so these are the advantages of high throughput sequencing. It has gotten much, 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 much cheaper. Okay, let's talk in general how we do this genome sequencing thing. Again, if there are any questions, please unmute yourself and ask, or you can put them in the chat if you don't have great audio, and I can stop and answer, because I can sometimes go on tangents about this stuff. I think it's really exciting. Okay, we're doing whole genome sequencing. We're starting with our purified genomic DNA, and it's got multiple copies, again, because every cell in your sample, every cell in the body, every cell in an organ has a full set of DNA, right? So in a sample, you're going to have multiple copies of all of the DNA. So this is showing you the chromosomes here for a little visual. And we're going to fragment them into small pieces, and this becomes our library. So our library are all of these pieces of DNA, and these fragments are all going to be simultaneously sequenced. So the machine doesn't just do like this piece and then this piece and then this piece. No, it does all of these pieces at the exact same time. And it says the order of the A's, T's, G's, and C's. Okay, and then what they do is they put those materials, those DNA molecules, over some sort of surface, usually some sort of microchip, but it can be like a thin gel, and they're going to separate those DNA molecules on a surface, and then they're going to sequence them from there, and a computer will align them. Okay, so on the surface, they're sequenced, and then those sequences are aligned based on overlaps. So what they do is they look for regions that have the same, and they're able to say, oh, this piece kind of goes over that piece, and together they get the full strand. So rather than sequencing this whole thing 1, 2, 3, 4, all the way down, they sequence these little chunks all at the same time and then use a computer program to align them. Okay, let's talk about all of them together, and then we'll go bit by bit. Okay, so this is an overview. Again, we will break this down further. This is what you guys, this is what we think about as the first revolution. So we call this shotgun sequencing. This was a huge advantage because we realized we didn't have to just go from one end of the strand to the other, right? We didn't have to go A, T, T, C, A, da, 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 da. No, we can break it up and sequence all of these pieces at the same time. That's called shotgunning. We're not talking about shotgunning a beer. We're talking about shotgunning a piece of DNA. Different. Okay, the first way that we did this whole genome shotgun approach took advantage of bacteria and plasmids. And so they would take each of these little pieces here and they would put them in plasmids and they would sequence them from the plasmids and then they would align them, okay? This is sequencing by synthesis. So again, we're detecting the addition of nucleotides, but we have to use some sort of bacterial system to make all these plasmids and grow them up, right? Make lots and lots of copies. And so it requires like a lot of fun with bacteria, which is labor intensive and takes up a lot of space and also kind of smells bad. It's got like fermenting bread smell, and it's a pain in the butt. And so then we have the second revolution where we realized that rather than using bacteria to do this step, we can do this on chips. And so for the second revolution, we're doing, again, sequencing by synthesis. So we're detecting the addition of new strands, but these are amplified not in bacteria. So we make lots and lots of copies not in bacteria. Instead, we make them on a chip. They're synthesis in vitro, so not in a live system, okay? Ooh. And the traditional way we do this is a company, Illumina Sequencing. They're the ones that do most of this type of second revolution high throughput sequencing, and we're going to watch a short video about Illumina sequencing. This is where we are now. We're in the third revolution, and what's cool about the third revolution is that we're able to do this at a single molecule level. So rather than having to take like a large sample to get this information, we can do this from just single molecule templates, and it's not as accurate, but it is giving us information about single cells, which is pretty interesting stuff in the science community. We're all really excited about that. And we'll look at PacBio and nanopore sequencing. We'll watch videos about them. So again, we started by having to use bacteria, labor intensive, smelly, slow. Then we use Illumina sequencing where we did this on a chip, and now we're doing it at a single molecule level. It's very exciting. Okay, we're gonna watch a video about Illumina sequencing, and this is showing you the machine. So it can fit like kind of on a bench top. UNCP actually has an Illumina sequencer. It's in the Health Science Center building, and it like fits on the tabletop. It's really cool. And this is the flow cell. So this is where the machine will kind of like flow your samples. And it's the size of like a microscope slide. So if you've ever taken microbiome and you've used those slides before, that's the size of where your sample goes. It's really small and it's got all this information on it. And the cool thing about an Illumina is that like robots inside the machine do all of the work for you. So you just like put your sample in and the robots move around and like pipette stuff. And it's, yeah, it's pretty new agey cool. Okay, so we'll watch a video about Illumina, then we'll watch a video about PacBio and Nanopore. Okay, so I'm gonna switch to YouTube. Are there any questions before I show these videos? Any questions yet? Okay, I know I'm nerding out a lot. I think this stuff is like really, really cool and exciting, and I know not everyone agrees with that, but hopefully the videos will help make sense of things. Okay, let me share my screen here and kind of pull this down. And again, if you have questions, feel free to unmute yourself and ask or put them in the chat. Leave me alone, YouTube. I do not want your service. Okay, we're gonna start. We're not gonna watch all of this about Illumina, but we're gonna start with some of this video just so that you get an idea about Illumina sequencing.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
Okay, so you take your sample and you don't actually do any of this. The machine, you add your sample in that you want to know what is the sequence of the sample, and the machine will add these little end pieces, right? And the end pieces, the indice is how it identifies what the sequence is. Like it gives all of them what amounts to a number. And then it also adds these little pieces at the end so it can attach to the flow cell. So it can attach to that slide. Here's the
is all of this happening at like a micro level?
Say that one more time, Anthony.
Is all this happening at like a very small level, like a very like small micro level?
Yeah, so the machine is the size of an oven. And all of this, you like open a side part of the oven and you add in your sample in a small test tube. And all of this stuff happens very, very small inside this machine.
Okay.
Fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with elan, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell.
Okay, so you take your little pieces of DNA, or the machine will take your pieces of DNA, and they've added these 2 regions on the end, shown here with the purple and the blue. And the purple will bind to the purple ones on the little slide, the little microchip, right? And they'll use a polymerase just like you would in normal PCR to make a copy. And then it's going to bend down just through dynamics and bind to a nearby blue section, okay?
Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments.
So you can take a small amount of DNA that you've chopped up and you've added, or the machine has added those purple and blue tags to, and when you add it to your slide, what's going to happen is through this process, you're going to make millions of copies of those fragments. So you're amplifying, you're making lots and lots and lots and lots and lots of copies of your fragments, and they're all attaching to this slide. So what you're doing in effect is covering the slide with your DNA.
After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template.
So again, with Sanger sequencing, it was termination, sequencing by chain termination. So we detected the end of the fragment. Here, we're detecting as each one is added, it's giving off a fluorescent signal that is specific for that nucleotide. So T's are one color, A's are one color, G's are one color, C's are another, okay? And it's detecting that.
After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read,
So again, what the machine sees is just like a bunch of lights going off, which I just think this is like the coolest thing. So all of these lights are going off, and the computer is able to read all of those lights. Okay, we're going to stop there. So it from there is able to give you a sequence, and then it puts it into a computer program, and it aligns all of those fragments, and it tells you with a fair amount of accuracy the sequence of your full piece of DNA. Okay.
So like in the video, it's like one strand, and then the other one comes through, and it starts like kind of reading it and sends off the light signals.
Yeah.
Is that how it also is in a machine? Like, so the computer can tell like which is which?
Exactly.
Or is it just millions of them going off at the same time?
Millions are going off at the same time, and the computer has like a camera function, so it's reading all of the different lights at the same time, and from there it's building a sequence in its computer brain. Good question. Yeah, so I think this is like the coolest thing, but it is a little confusing. Okay, so this is Illumina sequencing is the one that we have at UNCP. It's the most common type of next generation sequencing because it's the most cost effective, which is why we were able to afford it. And so in general, this is the one that's used. So we're going to talk briefly about smart sequencing, but it's not as common. Oh, the sound is being funny. Okay, so each smart cell contains tens of thousands of these tubes, these ZMWs, and they are a very small detection volume. So they're tiny, tiny, tiny. So you don't need to add a lot of sample to it. And they're going to be illuminated from below. So again, very, very small here. And then the light is going to just excite the bottom part of the well. And what it is a very small microscope, a zeptoliter, which is tiny, 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 tiny amount. Okay. And then your DNA template with polymerase is going to be bound to the bottom of your little teeny, very small well. So you've got a sequence here with a little polymerase. And then again, we have those fluorescent nucleotides that will be added to your tiny, tiny chamber. Again, each color representing an A, T, G, or C. They're labeled with a different fluorophore. So A is green, C is orange, G is yellow, T is like a pink color. And when they attach, you'll get a light pulse. So the thing will pulse yellow for the G, green for the A, and so on. And because it's such a small volume, it can detect that light really well because the well is so small. And so it's just going through, adding it, okay? Adding those little fluorophores, detecting that light. So it's the same sort of system as before, except rather than occurring on a slide, it occurs in these little teeny, teeny, teeny wells. Okay, the last thing I want to talk about is nanopore sequencing. This video works, thank God. Okay.
For DNA sequencing, intact DNA strands are processed by the nanopores and can be analysed in real time. The nanopore sequences the fragments that are presented to it regardless of their length, rather than generating reads of a specific length. This could be reads of hundreds of kilobases or more. Nanopore log...
Okay, so the difference here is that for the other ones, they had to break them up into small fragments, right? We saw that with the Illumina, they have to be kind of small fragments so that they can be bent over and form that little bridge. With a nanopore, it can be a very long piece of DNA, so you don't have to fragment your sample quite as much.
Long reads simplify assembly and sequencing of repetitive regions, also improving the speed of species identification in metagenomic experiments. So what happens during nanopore sequencing? The DNA strands to be sequenced are mixed with copies of a processive enzyme, shown here in green. As the DNA enzyme complexes approach the nanopore, the single-stranded DNA is pulled through the aperture. The enzyme ratchets the DNA strand through the nanopore one base at a time. The speed of the enzyme can be controlled. More data is yielded per second the faster the enzyme runs, but with no deterioration in accuracy. As the DNA moves through the pore, the combination of nucleotides in the strand being processed creates a characteristic disruption in the electrical current. This nanopore signal can be used to determine the order of bases on that DNA strand.
Okay, so a nanopore is a man-made structure and it has an electrical signal to it. And because of the different chemistry of an A versus a T versus a G versus a C and their different sizes, as that nucleotide moves through this tiny, 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 tiny pore, it will disrupt that electrical current and give you a voltage readout. And you can use that, or the computer picks that up and tells you the sequence in real time. So again, this is sequence by synthesis. It's telling you as it happens what is happening, okay? And the advantage here is you can do very long reads. Okay, I do not expect you to be able to tell me how all of these things work. Just throwing that out there. I just wanted to introduce you to the science behind these technologies so that when you read about them, you have a vague understanding of like what's happening. Okay, let me switch back to where we were. So, go back. Again, comparison of our sequencing platforms. First revolution, we're doing that Sanger shotgun sequencing. We're using bacterium. We saw this with our Illumina sequencing. This is the second revolution. We're sequencing by synthesis here. We have this flow cell, this little slide that's going to make these little bridges, and we'll sequence that. It's high accuracy, but again, the reads have to be short, so it can only do little fragments. So you really have to take your big piece of DNA and break it up into a lot of little pieces to use Illumina sequencing. In the third revolution, we saw the PacBio, right, with the little teeny wells, and the nanopore sequencing where it has that fake nanopore thing and it pulls it through. These, you can use single molecule templates, so you don't have to break it up into as many pieces, right? You can sequence kind of a whole long piece, and that long read length is helpful sometimes. Okay, so we saw these videos, great. With each run though, you're generating a ton of data. So you're generating billions of sequences here. And so you need to use lots of computing power to analyze this data. And so the limitation here is for most sequencing, each sequence read is going to be kind of short, 500 to 300 base pairs, except for those single molecule systems where our read length can be up to 50,000 base pairs, right? So that's an advantage of those nanopore and the PacBio systems. Okay, let's talk about money. So Sanger sequencing in bacteria is a lot cheaper. You have your template, you extend it, you arrange these little fragments, right? You detect them. But in general, you got to confirm all of this, and it takes a while. Next-gen sequencing, specifically the Illumina sequencing, is more expensive. It's not quite as expensive as PacBio or a nanopore, but you get like a lot more information out of it. So you're going to get the whole genome, you can do your whole transcriptome, you can do metagenomics, right? And it's all done on these chips, so it's a lot faster. So it depends on how much money you have and how much time you have. So I'm going to break you guys into groups, and all I want you to do is just talk about what you got about the different sequencing technology and answer the question, if you were designing an experiment, which one would you use? So we're going to do like a very quick 3-minute discussion. What did you learn from the first part of class? Did you learn anything? And what would you pick as your sequencing technology? So I'm going to break you guys into breakout rooms here. So go into your room and unmute yourself and just talk about what you learned. Did you learn anything? What would you pick? Khalil, are you okay? Khalil, are you here?